The session that I chaired gives us a glimpse into the future. There's a number of gene therapies, the current state of the art based on the AAV vectors. Two of them have been approved recently, and they represent a tremendous progress that has happened in the field of hemophilia treatment. But they have several imperfections, and future iterations of gene therapy will try to solve some of those problems. And we've seen in the session several such incremental improvements of what the current therapies offer. But having said that, they have a long way to go before they demonstrate their potential in the clinic. These are preclinical studies. They each solve one or two problems of the current gene therapies, but none of them solve all of the problems. So we will have to wait for something that will provide a true cure in the sense that the community has been awaiting for decades. Among the problems that limit the current gene therapies, those approved and several others that are well on their way to the market, are problems such as gene dilution effect, which prevents us from delivering these therapies to children, as well as wide variability and unpredictability of efficacy. Those new generations of gene therapies will be able to solve some of those problems, which hopefully ultimately will lead us to that holy grail that the community is awaiting. For example, some of the approaches presented during the session may permit redosing or even titering the dose to achieve a desired expression level, which may minimize the problem of variability and unpredictability of the therapy. But these are non-viral vector approaches that I'm talking about that will be able to solve those problems. But until we are able to do that, the AAV vectors are here to stay, despite their limitations, and there's room for improvement of these therapies. And we've also seen some potential for that during the session. There is potential to improve expression levels by playing around with the coding sequence for the proteins, such as coagulation factor 8, which is an extremely challenging protein to make for any cell whatsoever. And hepatocyte is not the natural site of expression of factor 8 in the body, and therefore it might be even more challenging for that cell to make it. And so making it easier for the cell might make those therapies that much better in the future. So in that sense, I think we'll see incremental improvements in the way gene therapies can deliver on the promise of providing the cure to patients.